Litter spread by people poses a great problem in modern society, especially plastic litter. People are aware of this widespread problem, and much research has been done on the effects of plastic waste in ecosystems. But what is the effect of plastic on a smaller scale in a world of terrestrial invertebrates hiding in the land down under? Many terrestrial invertebrates need shelter sites in order to survive. Shelters provide a set of services that improve an organism's chance of survival. These services include favorable microclimatic conditions, for example low temperature and high humidity, protection from harsh conditions, for example hard winds and heavy rainfall, shelter from predation, and an offer of additional resources such as food. To terrestrial invertebrates, plastic litter spread across their habitat might provide some of these services and may thus also function as a shelter. Different shelter types, such as wood, stone, or plastic, each offer their own set of services, which affects the diversity of organisms using it as a shelter. Plastic. A plastic object provides shelter from predators and from harsh weather conditions. However, it does not provide food, it is not edible, or favorable microclimatic conditions. It heats up very easy. Stone. Stone objects provide shelter from predators and from harsh weather conditions, and provide favorable microclimatic conditions as it provides a cool and humid environment. However, just as plastic, it is not edible. Wood. Wood provides shelter from predators and from harsh weather conditions. It has favorable microclimatic conditions and additionally serves as a food source. Different combinations of services attract different organisms. This, in combination with competition between species, will lead to the observed species composition underneath various shelter types. This led us to ask the following research question. Which shelter type, wood, stone, or plastic? To test our hypothesis, we performed 3 studies: a correlative field study, a manipulative field study, and a manipulative lab study. We selected 3 beech forests to serve as locations for the correlative and manipulative field studies. We chose beech forests as they are uniform in tree composition and have little undergrowth, reducing the amount of confounding factors and offering an accessible area. Other confounding factors we measured were soil temperature, soil moisture fraction, thickness of the leaf litter layer, surface area of the shelter, and the weight of the shelter. Soil temperature and soil water fraction were measured as these are factors describing the microclimatic condition beneath the shelter. We believe that microclimate plays an important role in shelter selection. Thickness of the leaf litter layer is a confounding factor as we believe it affects the present habitat size and thus the abundance of terrestrial invertebrates. Shelter surface area was measured to correct for the fact that a larger area will have a higher chance of containing higher richness and abundance. Shelter weight affects the stability of a shelter. We expect that shelters which are more stable to contain a higher terrestrial invertebrate abundance and richness. In a pilot study performed in advance of this study, we found that plastic and stone shelters were far less abundant than wood shelters. The optimal situation would be to find the 3 shelter types close together, however, this rarely occurred. We then decided to first search for plastic and stone shelters and then look for a wood shelter in the immediate vicinity. When we found multiple wood shelters nearby, one piece was randomly selected by allocating numbers and randomly selecting a number. After a shelter was turned over, we collected all invertebrates on the soil surface directly underneath the object or on the surface of the object. Immediately after the invertebrates were collected, the soil temperature was measured, and a soil sample was taken to later determine the soil water fraction. We then measured the thickness of the litter layer, the surface area of the shelter, and its weight. Lastly, the invertebrates were identified to order level, and the number of individuals per order was recorded.
To test if the chosen sample size was large enough, we performed a post-hoc power analysis. By looking at the intersect of the graph with the 0.05 line, we can see that a minimum of 22 samples were needed. As we have 99 samples, we are far above this value. To analyze the data of our correlative field study, we used a generalized linear model with a log-linear binomial distribution. As dependent variable, we used order presence, as main factor shelter type, and as random factor day. In total there are 66 replicates for wood, 16 for stone and 17 for plastic. As a reminder, our expectations were that the highest abundance and richness would be found underneath wood shelters. We will now show a selection of the results which showed a significant effect of shelter type. Two of the orders not discussed here had p-values between 0.071 and 0.05. In this graph you can see the percentage of shelters where isopoda were present on the vertical axis set out against shelter type on the horizontal axis. Isopods show the highest preference for plastic, followed by wood. When analyzing the data for Diplopoda, wood and plastic shelters seem to be equally favored, and stone the least. When studying the results of presence tests, there seemed to be a general preference for plastic. Stone shelters overall are found the least attractive. However, considering the low amount of replicates for stone and plastic, the tests might not provide reliable results. There is a high possibility that shelters are just randomly selected, as many of our results did not show a significant effect of shelter type. When comparing our results with the findings of SANS, who did research on the abundance and diversity of invertebrates in wood and PVC stacks, we find similar results. As can be seen in this graph, SANS found a lower invertebrate abundance in plastic shelters. However, when looking at the diversity, plastic is equal to wood. We also performed a general linear model and a Kruskal-Wallis test to analyze the effect of shelter type on abundance. These tests were left out of the results as they were not representative because of the low number of replicates of stone and plastic, as many had a value of 0 and could thus not be used. Our manipulative field study consisted of placing stone, wood and plastic shelters in beech forests and determining the richness and abundance of terrestrial invertebrates using them as shelters. On each location we randomly laid out 11 blocks, each block containing the 3 shelter types. We chose 11 blocks per site as we were limited by the 100 pitfall traps that were available. Here you can see a schematic representation of such a block. The 3 shelters were placed in a triangle, each 2 meters apart. This distance was chosen to keep the shelters further apart than the daily action radius of terrestrial invertebrates. Once such a triangle was laid out, the corners were given numbers and the shelter type was randomly assigned to each of the corners. Then the soil temperature was measured and the hole was drilled with an auger. On the edge of the hole, the depth of the leaf litter layer was measured. We did not take samples for the determination of soil moisture content, as it was logistically impossible to dry and weigh an additional 198 samples each day. A pitfall trap was placed in each hole, filled with a small amount of 70% ethanol and covered with a 15 by 15 piece of the assigned shelter type. Shelter size and weight were not measured, as these were controlled by creating shelters of identical size. The weight only varied between shelter types. This setup was then left for 24 hours. The following day, we returned to empty the pitfall traps, measure the temperature and relocate all traps at different locations. This procedure was performed 3 times, resulting in 33 replicates per shelter type per site. For this experiment we also performed a power analysis, using the same procedures as with the correlative study. This time the interception is at 60 samples, and with 297 samples we are again far above this value. For this experiment we also performed a power analysis, using the same procedures as with the correlative study. This time the interception is at 60 samples, and with 297 samples we are again far above this value. To analyze the results of the manipulative field study, we used a generalized linear model with a Poisson log-linear distribution. We used the Poisson distribution as we used counted data. The data of our manipulative field study contain many outliers, resulting in a less reliable statistical analysis. The dependent variable is the number of organisms. As main factor we use shelter type. The random factors are day and location. And the covariates are temperature and the depth of the leaf litter layer. The number of isopoda individuals which were found during the manipulative study was low, whereas isopods were the most abundant order during the correlative study. It is possible that isopoda can smell alcohol in pitfall traps and therefore stay away. This graph shows the abundance of diptera in the vertical axis set out against shelter type on the horizontal axis. In our manipulative field study, contrary to our correlative study, we found a high abundance of diptera. Diptera show a significant preference for shelter type. They prefer plastic over stone and wood. Wood seems to be their least favorite shelter. The effect of shelter type for pulmonata is significant. From the p-values we can assume that plastic is not significantly different from stone, yet the graph implies that it does. This could be caused by the high amount of outliers. In this graph, the order richness is set out against the shelter type. The effect of shelter type on the order richness is significant. Plastic has a significantly higher value than stone. Stone and wood harbor similar order richness. The temperatures beneath the shelter types do not differ significantly from each other. We expected to see a difference, which we assumed to be one of the major factors for shelter selection. However, this was not found. This implicates that the 24-hour time period was probably too short for the microclimatic conditions to differentiate between shelter types. Our lab study consists of a series of preference experiments used to test which shelter type was favored by our test subjects. For this experiment, we used organisms from the 4 most abundant orders we found during our pilot study. We chose the most abundant orders as large amounts of subjects were needed to perform all treatments. The selected orders also form a diverse selection from the orders present in beech forests. The orders we used were Isopoda, which are moderately moisture-dependent detritus feeders, Chilopoda, which are carnivores, Haplotaxida, which are highly moisture-dependent detritus feeders, and Pulmonata, which are herbivores. The arena we used consisted of a small plastic box with a stone, plastic and wooden shelter, each of 5 by 5 centimeters. These shelters were then randomly allocated to one of the 3 locations within the arena. 80 separate arenas were created to be able to perform all treatments within the given time. In total, we performed 3 preference experiments: a single organism preference test, a multiple organism from the same order preference test, and a multiple organism from each of the 4 orders interaction test. Single organism preference test was performed by randomly selecting one animal from the total caught animals of one order and placing it in an arena. This was continued until 20 individuals of each order were in separate arenas. An equal amount of replicates for each order was chosen to account for the possible differences in sampling day. The arenas were subsequently left for 24 hours. The next day, the hiding locations of the organisms were recorded, resulting in a preference score for stone, wood, plastic, or no choice. This was repeated 2 times for a total of 40 replicates per order. The multiple organisms from the same order test was performed using the same procedures as the single organism test. However, this time 5 individuals of the same order were placed in an arena. This experiment was replicated 17 times for Chilopoda, 20 times for Isopoda, 13 times for Haplotaxida, and 13 times for Pulmonata. The amount of replicates was limited by the amount of individuals available per order. The organisms used in the single organism test were reused for this experiment. The multiple organism from all four orders test also followed the same procedure as the previous two experiments, except that here 2 individuals of each of the 4 orders were placed in the same arena. This test was replicated 30 times. To analyze the data of the lab study, we used the chi-square test, combined with the chi-square test of residuals, which is similar to a post-hoc test. As main factor we chose shelter type, and as dependence variable we chose number of individuals. No random factors or covariates were chosen, as they were all controlled for. In these graphs, number of individuals is set out against shelter type. Each time the single individual experiment is shown together with the group experiment, to compare for aggregation behavior. In the single individual test, the Chilopoda did not show a significant preference. Once they were placed in a group, they showed a preference for stone. This might be due to aggregation. Isopoda show a similar preference for plastic in both single and group experiments. Comparable to Chilopoda, the Haplotaxida did not show a preference in the single individual experiment, but did show a preference for stone in the group experiment. The preference of Pulmonata seems similar between individual and group experiments. However, the group experiments show no significant difference where the individual experiments do. When comparing the results of the interaction test with the individual and group test, we can observe similar patterns between the interaction and group experiments for each order. This would imply that there's no clear difference between the effect of multiple individuals of the same order or multiple individuals of different orders. This seems strange, as most interaction experiments resulted in a massacre of the other orders by Chilopoda. This would suggest that there are plenty reasons to want to avoid being predated upon by Chilopoda. However, there seems to be no support of avoidance behavior when comparing the results. Lastly, we will offer some general conclusions. From the correlative field study, we can conclude that most tests do not show a significant effect of shelter type, which is probably caused by the lack of replicates for stone and plastic shelter types. And without the abundance test, it is hard to draw any concrete conclusions on the preference of terrestrial invertebrates. To improve this study, more replicates of stone and plastic are needed to improve the results. For the manipulative field study, we can conclude that the order richness seems to be highest underneath plastic shelters. However, under the circumstances of this experiment, there are no differences between soil temperature underneath the 3 shelter types. For the manipulative field study, we can conclude that the order richness seems to be highest underneath plastic shelters. However, under the circumstances of this experiment, there are no differences between soil temperature underneath the 3 shelter types. As this was one of our hypothesized driving factors for shelter selection, this supports the idea of random shelter selection. We can also conclude that, due to the high amount of outliers in the preference tests, we cannot draw any hard conclusions from this data. For the manipulative lab experiment, we can conclude that this experiment yielded the best quality data out of our 3 experiments. Therefore, we can observe that there is no consistent pattern in shelter preference between the 4 different orders. We can also conclude that there is probably no competition effect of multiple orders in one arena, as the results of this test were similar to the results of the 5 individuals of the same order test.
Weet je wat ik zie als ik gedronken heb? Allemaal beestjes. Zoveel beestjes om me heen. Oh, ik weet wel dat ik nou mezelf nep. Want er zijn geen beestjes. Maar ik zie beestjes om me heen.